Before we carry on with mitosis and what details will be included in that process, I want to make sure, once again, that you're paying attention to the vocabulary here and noting the actual differences between some of these terms that probably sound the same. Chromatin is loosely condensed, or I should say uncondensed DNA, loosely arranged DNA. DNA is really read when it's accessible, remember, by RNA polymerase and transcription factors that can bind to the promoters, etc. And so DNA is typically present as loosely arranged chromatin. When it becomes condensed around, really tightly organized, well organized around histone proteins and condensed into chromosomes, you use that term. Remember, chromatids, as we just discussed in the previous video, are simply the duplicated chromosomes that are held together at the centromere. And so if you look at a diagram of chromosomes, you'll see that you've got sister chromatids held together at the centromere, but a chromatid is really one of the two chromosome copies for a very particular chromosome. And then centrosomes and centrioles, these are not related directly to the chromosomes. These are features of the cell that are involved with the development of the mitotic spindle. And so the centrosome is the region in which centrioles occur, and you've got these organizing units then producing microtubules that extend from those regions to form what's called the mitotic spindle. And here, if we look at this arrangement of chromosomes, we'll see you should see there are 30 pairs. You'll notice that 1 through 29 are all labeled with numbers, and that this last pair is labeled X and Y. Well, with chromosomes, first of all, this arrangement is called a karyotype. They don't occur in the cell this way naturally. It would be more of a chromosome soup in terms of their arrangement. But we can identify and organize and label chromosomes from cultured cells, producing kind of a map of chromosomes called a karyotype. And so this is the karyotype for a bull. Humans actually have 23 pairs, and again, that would be a total of 46 if you looked at a human karyotype. Regardless, here 1 through 29 or 1 through 22 in us would be what are called autosomes. All we're distinguishing with the use of autosomes and sex chromosomes is that final pair is the sex-determining pair, therefore called sex chromosomes. So there are 30 pairs here in this karyotype, 29 are autosomes, and then we have one pair of sex chromosomes. If we look at this one, we've got 36. Here I've already included the fact that this is one from a dog, and so different species have different numbers of chromosomes in their cells. Humans have 23 pairs. Here, when it comes to dogs, they actually, if I move the dog out of the way here, if I can, no, I've got it grouped. They have a different number of chromosomes. And then here, of course, when we look at where all this is at, we're focused on the nucleus when you're talking about eukaryotic organisms. But take a look at these features here. We've got a centrosome with centrioles. This is a centriole here. Those are the structures that are going to give rise to the mitotic spindle. They also will be duplicated prior to division so that we have two. One is going to be on each pole, each end of the cell, and separate the chromosomes. And so here you've got a pair of centrioles within a centrosome. And again, that's putting it here with this figure in the context of the cell itself. These microtubules will extend to create the spindle fibers. I do want you to realize there are different kinds of spindle fibers. There are kinetochore microtubules that attach to the protein complexes of the chromosomes themselves at their centromere, where the chromatids eventually will be separated. Remember, we want this copy of the DNA, this set over here. We want these chromosomes, the copies of these. We want these 3 chromosomes to head over here. So in the end, we can form 2 daughter cells, each of which has a full set of the DNA. But here we also, you'll notice in tan, we have these other microtubules extending from the centrosomes. These are called polar microtubules, and they're actually elongating the cell. It should make sense to you that elongating the cell, stretching it out by pushing these microtubules apart, actually makes for a safer division between the DNA that will be transferred over here and the DNA that will be shuffled over here. And then we've got these anchoring organelles called astral microtubules. I will probably not ask you about these, but I'm interested in you knowing about the kinetochore microtubules and the polar microtubules. And so if we continue to take a look at that, here you're showing all three: the astral microtubules, polar microtubules, and kinetochore microtubules. We don't need to worry about the plus and minus ends. That's something that we can explore in AP Biology, or you can, of course, ask me in person. But it has to do with where those microtubules are being elongated or shortened. And these microtubules are not pulling the chromosomes apart. What happens is they are shortening on this end eventually, and as the kinetochore here is linked with this microtubule and it shortens at that spot, this chromatid, this sister chromatid, will head this way. This sister chromatid will head this way. Here, there's more information on what goes on with the copying and kind of the cycle of the centrosomes. Please do not feel at all like you need to memorize this, because that is absolutely not the case. When we look at these again, we've got two pairs of chromosomes. Here's one pair. You can tell by the length that those go together in this diagram. And then you've got a maternal and paternal copied chromosome each. Looks like squid here with these little eyes. Those dots are actually, again, the kinetochores. They're present at the centromere. The centromere is where the sister chromatids are attached. But again, the color, the difference in color, red versus blue, indicates maternal versus paternal. And the difference in size suggests that, hey, we've got a different chromosome here. These two go together as maternal and paternal for one pair. This is another pair, but each has been duplicated. This is an actual transmission electron microscope image showing those microtubules and then showing kinetochores. It's difficult to make out much more in that particular view. Then again, with interphase, that's most, if we jump back to interphase, that's most of the cell cycle. We've already covered this, so we'll go on. But again, the chromatin in prophase, when we start looking at mitosis and the stages, the chromatin is becoming tightly coiled. The chromosomes are not arranged in any particular fashion here, so that the fact that you can see chromosomes and the beginning of a mitotic spindle, but the fact that you can see the chromosomes indicates that we're in prophase, because those chromosomes are not arranged in any particular way. That's further evidence that this is probably prophase. And then what's going on is the nucleoli disappear, the nuclear envelope begins to degrade, the centrosomes are beginning to migrate to opposite poles of the cell, and mitotic spindles beginning to form. If we look at prometaphase, that's kind of intermediate. The nuclear envelope is really broken down. There are no nucleoli. We see that the chromosomes are attached to the mitotic spindle at the kinetochores, which again are positioned at the centromere of the chromosome. And once we get to metaphase, we see that, hey, those chromosomes are actually aligned, single file here, on the equatorial plate or metaphase plate, as it is also called. The mitotic spindle is well developed, extending from centromeres and centrioles that are on opposite poles of the cell. So essentially, we're all lined up, ready to divide. This is the hallmark of metaphase. And then when we look at anaphase, the metaphase takes a while typically, but anaphase is typically quite short because it's all ready to go. What has to happen is simply that these microtubules, these kinetochore microtubules, shorten, separating the sister chromatids at the centromere and drawing them to opposite poles. And so again, this is an exact duplicate of this one over here. So when it reaches this end of the cell and this one reaches this end of the cell, and eventually we divide them down the middle, the DNA that is in the daughter cell that will be over here and the DNA that is in the daughter cell that will be over here will be identical. We'll have just two new cells of exactly the same kind with exactly the full set of the same DNA. And so the cell's elongating a little bit with those polar microtubules not shown here as well during anaphase. And then if we look at telophase, again, with telophase, we are starting to get a cleavage furrow. If this is an animal cell, there would be a ring of actin protein here that is getting shorter and shorter, or smaller and smaller in diameter, essentially pinching off these two cells from one another. In telophase, you can see that the chromatids have been separated onto opposite ends of the cell. Now they are called chromosomes. Again, sorry if that's confusing, but that's the way it works. And then the daughter nuclei are reforming. We're actually starting to see a nuclear envelope reform. And so we will end up eventually, and you can see there's only one centrosome now with one pair of centrioles. So if you look back at that cycle that I showed you on the slide a little bit earlier, for the duplication of centrioles, you'll see what stage we're in here. No need to do that. You won't be tested on the centromere cycle whatsoever. But this indentation here is called a cleavage furrow, and again, as this ring constricts, it'll pinch the cells off into two. And then if you look back, you'll see that this is exactly what the parent cell started as, with just two single chromosomes, maternal and paternal, for each of these two pairs. Once more, I wanted to point out that we're only following two pairs of chromosomes, because to follow 23 or more would be rather heinous visually. It would be too complicated. And here, this further emphasizes what's going on with cytokinesis. You can actually see the division of the cytoplasm occurring as we form a cleavage furrow with this actin ring, and that leads you to the two daughter cells that you would have. Remember that with plant cells, you've got vesicles fusing to make a cell plate, so the process is a little bit different depending on what lineage you're talking about, what type of cell you're talking about. What's highlighted most often would be the difference that you see between plants and animals. This is supposed to represent the actin ring between these two fried eggs here. Of course, those represent the different daughter cells. Here, the brown would be the new daughter nucleus. And then if we look at what we were supposed to see with the microscope, the onion root tip, this is the tissue of the root. So you can see the individual cells. This is pretty cool here. In my view anyway, you can actually see these little purple dots represent the nuclei. So that's the nucleus of the cell. We have to really zoom in, though, on this tissue to see what's going on in those nuclei, and that's what this figure is. And so you can see these kind of yellow-brown borders here. Those are cell walls. And then you can see these stained circles here where it is even, where you just see it's kind of grainy and you don't make out individual, sausage-shaped chromosomes. You've got the cell in interphase. But here, of course, we can see a very distinct alignment of chromosomes in the middle of the cell. What is that stage? Of course, metaphase. Here's another one in metaphase over here. Another one in metaphase over here. Where would prophase be if we're starting out? Well, you can see that there are chromosomes. It's not as even and grainy as this nucleus here. This one's in interphase, but next door you can actually see the chromosomes. They just don't have that particular arrangement. They're kind of all over the place, and so that's clearly prophase. This is clearly prophase in the upper right. And again, when you go from prophase to metaphase, you've got an alignment of the chromosomes in the middle. So these are very clearly in metaphase. And then if you see here, within this one box, this one cell, those chromosomes are being separated. And so this is anaphase, this is anaphase. This one's a very nice example of anaphase. And then here, this is somewhere between anaphase and telophase, because you've got the chromosomes really well separated. And it's within one cell. So if nothing else, you know that telophase is going to be occurring just after this point in that cell. And then you might see some with daughter nuclei. Here are narrower cells. It looks like these probably just underwent cytokinesis and divided into the two daughters. And so when you look at these cells, you can identify those separate stages, and that might be of interest. And so once more, if we just look at an overview for mitosis, keep in mind that in interphase, we really don't have chromosomes apparent, even though most of the figures will suggest that we do. That's just so you can trace things visually and kind of understand what's going on. But the DNA would be present as chromatin at this stage, and it's really in prophase that we see the chromosomes and we see that they're already duplicated. Why? Because that happened in interphase during S phase. Then in late prophase, they're starting to get shuffled. Eventually in metaphase, they're aligned at the middle along the metaphase plate because of the action of the spindle fibers that are attached to them. In anaphase, they separate. In telophase, they have been drawn to opposite ends of the cell. We get daughter nuclei forming, and eventually cytokinesis divides the cells so that this cell here has the exact same complement of DNA as this one over here, the other daughter, and each one is identical, unless there's been some sort of mutation, identical to the parent cell. Mitosis is just a mechanism for copying cells, making two cells from one.